Okay, that's fine. Thank you. We're just gonna ask you to stand here because otherwise you can see it in the camera. Yes, and then you'll control this. Yes. Yes. Thank you. Yes, you can start.
I don't know. I mean,
Yeah, I think we're good.
and then he's throwing up.
Hey everybody, welcome to Tropilunch. Today we have a great speaker with us. I'm going to introduce him. His name is Lungelo Khanyile. He's a second year student in the PhD program in the biology department here at the web. And he's under the supervision of Dr. Nico Solnit. He holds a master's degree in agricultural biotechnology from RCS University in Turkey. His research interests focus in the application of bioinformatics and phylogenomic analysis to investigate the evolutionary trajectories of alloptogenous and autochthonous endemic lineages in Africa. For his doctoral research, he uses Monopsis from the Campanulaceae family as a model clade to reconstruct the evolutionary history of African flora. In collaboration with researchers from the South African National Biodiversity Institute, SAMBI, the University of Swatini, and the Swatini Herbarium. Without further ado, let's welcome Lungelo. Great.
Thank you so much for this opportunity. I'm Lungelo, as you Erin said, I'm from Eswatini. How many of you guys know Eswatini? Such a good number. Yeah, from Eswatini, like a small country like in Southern Africa. It is not Lesotho. I want to clarify that because a lot of people end up saying like we are Lesotho. Lesotho is inside South Africa, but like we are between like South Africa and Mozambique. So basically, today I'll be talking about my research, which is based in Sub-Saharan Africa, but a majority like, of my species they occur in Southern Africa, but we've got like few lineages that are occurring like in Western Africa and Eastern Africa and then like Central Africa down towards the south. So that's why I'm here, and I hope you guys are going to help me to like answer some of the research questions that I have, because I'm still like in my second year, so I'm still developing some ideas. But I've already like started like working on my research, but I'm here like to try and like share what I have right now, and then also to get more ideas. Hopefully, I'll be able to develop like a better like research to actually like answer more questions when it comes to evolutionary history, because currently like this ongoing climate change and global warming, which actually affects like biodiversity in Africa, even in the rest of the world. So my research is primarily about that. And then, as you already said, I'm in the Department of Biology, but I'm also like associated with the Florida Museum of Natural History. So I'm just between those two departments, if I can say. Without much of wasting your time, I'll just dive into my like presentation and go on.
Hide the camera button. There you go. Now you can.
Oh, okay, finally. So just like briefly like explain or talk about the background of my research. So like since I'm from Africa, I'm just like familiar with like the ecosystems in Africa. And Africa is rich, not in terms of economy, but in terms of like biodiversity, because we have a lot of biodiversity hotspots. I'm from Swaziland, so there's a biodiversity hotspot next to Swaziland. It is called like Maputaland, and then there's also like the Cape Touristic region. I think if you're working in Africa, you're familiar with the Cape Touristic region because it's one of the most species-rich like biodiversity hotspots in Africa. And then like there's Horn of Africa, which is in East Africa, and then there's also like the one in Western Africa, just along the Cameroon volcanic line. So basically, like this rich biodiversity, we assume that like it was caused because like after the break of the Gondwana land, because I think you guys are familiar with the phenomenon that about 100 million years ago, South America, Africa, and then like Asia together as one continent called the Gondwana supercontinent. And then after that break, which happened about like 100 million years ago, Africa continued to evolve like independent, whereby like we had like a lot of both climatic and geological events, such as like the continent was able to tilt 15 degrees east, and then there were like Bengal currents, and then like the uplift of escarpments and continuous volcanic eruptions, especially like in Cameroon. So we assume that, or we know that like these events might have shaped like the current biodiversity or the current pattern of distribution of both like flora and fauna. But we are not yet sure on how like these different events actually influenced the current distribution of biodiversity. So basically, like that's what we're trying to understand, to try and actually like correlate these like geological and climatic events with like speciation events, so that we can know how particular like climatic or geological events influences or affects like the distribution of species that we see in Africa. And then like, why am I doing this? I'm doing this because currently like the rich like biodiversity in Africa is facing a major challenge, because we're losing like our biodiversity because of climate changes, land use changes, and many other like factors. So like in the past, I think like 20 or 30 years, like according to this research, like Africa has lost like 70, like projected like a 76% loss of its biodiversity, which is huge, considering that like some of the species are just endemic to Africa, which means basically like just like found in Africa only and not anywhere else in the world. So like if we lose like these lineages and these species, chances are like the ecosystem is not going to be stable in the near future. So in order to try and understand like species that are at risk of being extinct, we had to actually like understand the evolutionary history, so that we can be able to infer like conservational strategies and try to conserve species that are at risk, so that we can have like a stable ecosystem. So like basically like my main idea is to actually like understand how the past events influenced like the current distribution, and try to predict how future like projected climatic changes and are going to affect the current distribution, so that I can be able to like infer like conservation strategies. So we'll do this by actually identifying like evolutionary significant units. Basically, like we've been trying to identify like the different populations within the African ecosystems, and try to find like genetically isolated populations which might be at risk of extinction. And then also we'll try to find like particular like niches or particular like refuge that are at risk, or that are going to be affected by the ongoing like maybe climatic changes, which might lead to some species ranges contracting. And then like in that case, once you identify like those species and those like ranges that are at risk, you might be able to actually like infer conservation strategies and see how we can be able to check like conserve some of the species and some of the ecosystem. And then like how do I do that? Basically, like we're going to use like a model plant, which is like Monopsis, and then Monopsis falls under the Campanulaceae family, which is like a big family, because it consists of about like 2,400 species. And then like this family is divided into 5 subfamilies. There's like Campanulaceae, and then like there's Cyphoidea, then Lopeloides, Vocabia, and then Nemacloides. Campanulaceae is one of the most studied like subfamily in my lab, because my advisor, basically like she's the queen of this family. So she knows everything. So like yeah, and then like it is distributed like we do have like African lineages such as Tropilunch and Beja and Flora. And then like Cyphoidea, it's endemic to Africa. So that's why like Africa is very important. If like the whole family is just like endemic to Africa. So imagine if we lose like a whole family because of the ongoing like climatic changes. That's huge. So and then like we've got Cappadocia, which is like in northern Chile. I've never been there, but like I think we've got few species of that. And then we've got Nemacloides, which is found here in America and then like in Mexico. And then lastly but not least, we've got the most important subfamily, which is Lobeloides. And this is important because like my model genus is actually like under this subfamily. It is the largest in the group because it consists of like 1,200 species, and it is like distributed in the southern hemisphere. of course, when we talk about the southern hemisphere, we're gonna talk about like Africa, and then like it is found most like in the near tropics. And then like my model plant, which is like Monopsis, is under this subfamily. And then like lobeloidy, it's a good like model to use because it consists of like diverse growth forms. We've got like big lobeloids, which are found like in the Hawaii region, and then like also in East Africa. And then we also got like small herbaceous plants, and Monopsis is one of the small herbaceous plants. So since they've got like a diverse range of like growth forms, it means that like they occupy different ecosystems. So like instead of like from the Mediterranean region to tropical forest to alpine like ecosystems. So basically like they're just found like in every ecosystem. And then like we do have like many like lineages in Africa, such as Monopsis, Campanulaceae, Umbrella unigens, and Lobelia. Some of those like are just like endemic to Africa, like Monopsis and Campanulaceae. They're just like found in Africa only. So, and then now I'm gonna talk about Monopsis only, and then see how it goes. So I decided to choose like Monopsis as my model because I feel like it just like gives like a full representation of the disjunct like populations in Africa. Because like Monopsis is found like in all like the major biodiversity hotspots in Africa, such as like the Cape Floristic region, then Horn of Africa, Cameroon volcanic light, and also like the Sakhalin region, and also like the Maputo like biodiversity hotspots. So to actually like use these plants as like as a model, it's actually going to help to actually understand the evolutionary history of African flora, because like we are going to be able to actually like predict and hypothesize how like sub-Saharan like flora actually like evolved, because it is not just like Monopsis which is just distributed like this. We've also got like other like plant species such as Disa, and then also do also have like animals like which also like follows the same distribution pattern, such as automs, which are like small mouse. And then like basically like to actually understand like the evolutionary history is not only going to help us understand about the evolutionary history of just like Monopsis, but it's also going to help us to develop hypotheses that can be tested in other like plants and even in other animals. And that way we are going to be able to better understand the evolutionary history of sub-Saharan like African flora. And then also the main reason it's because like it is found in events or in locations whereby like major climatic and geological events happen. Because like in Cameroon, like in the Western African population, like a lot of volcanic eruptions happened in the last like 30 million years. And then like in East Africa, like if you follow like African geography, they're like the uplift of escarpments, and then like we hope or assume that they might have influenced like the distribution of African flora. And then like in the Cape region, like there was a Benguela current which actually like helped to form like the Mediterranean region in Western Cape. And then like to actually like understand how like these different geological and climatic events influence or shape the ecosystem is going to be very important for us, so that we can understand how future climatic changes might affect or influence this distribution that we see. And then like we are going to do this using 3 hypotheses. But before like I talk about my hypothesis, like those who work in Africa, we've got like 2 major hypotheses in Africa, which is called like the Cape to Cairo, and then there's also the one called like rain flora like hypothesis. So the Cape to Cairo basically is like, if you know, Cape Town is in Southern Africa and then Cairo is Northern Africa. So the Cape to Cairo like hypothesis basically like means that like there was in situ like radiation in the Cape region, and then later on like the species they migrated northwards towards like the Cairo region. So it's like in situ radiation and then like northwards. And then like the rain flora like basically like assumes that million years ago like there was a uniform ecosystem in Africa, and then after that there was eradication in all the other like climatic events, which actually like caused like fragmentation of the ecosystem. In that way, like vicariance was created in such a way that like we end up with like the relevant or different like subpopulations like in Western, Eastern, and then like in Southern Africa. So those are the 2 like major hypotheses that we usually have, or that we usually like have used to actually like describe like the distribution of both like flora and fauna in Africa. But like my hypothesis is quite like different because like my hypothesis number one basically like is almost the direct opposite of the Cape to Cario because like I assume that like Monopsis originated in the tropical forest, which is somehow northwards, and then like about 17 million years ago during the oscillation of climate temperatures, when like the tropical forest started to fragment, we assume that that's when actually like Monopsis was able to jump from the tropical forest to the savanna ecosystem. So in that way, it was able to colonize like a new ecosystem. So that's my hypothesis. So basically, we're just trying to understand if Monopsis like originated in the tropical forest. And then hypothesis number two, basically like we've got like escarpments in Africa, Western, Southern, and then Eastern Africa. So these escarpments, like sometimes they act as corridors of inflow, sometimes they act as barriers of inflow. But like in our hypothesis, we assume that like since, according to our hypothesis number one, like Monopsis originated in the tropical forest. So we assume that like during the uplift of the Western like escarpments in Angola, they added like the colonization of Monopsis from the savanna and from the tropical forest towards like the succulent region. The succulent region is one of the arid like ecosystems in Africa. It's found in Namibia, Angola, especially like in Western Africa. And then that is hypothesis number two. And then hypothesis number three, we then assume that like since now the Monopsis was able to colonize the dry ecosystem, it then like jumped into the Mediterranean region in Cape Town, like recently. We just assume that like this is a recent event, like late Miocene to Pliocene. So those are the three hypotheses that I will be testing like in my research. And then like I will test this hypothesis using like three objectives, whereby objective one, I'll just try to reconstruct the evolutionary history of Monopsis and Lungelo. And then in that way, I will be able to actually like understand like the relationship between like the species and then like within the family. And then like objective number two, whereby like I'll try to actually like date the divergence at times so that I can be able to correlate like divergence or speciation events with major climatic and geological events so that we can be able to assume or hypothesize that if, for example, like there was a Benguela current 7 million years ago, and then we see in our phylogeny that like about 7 million years ago, like that's when like a lot of divergence happened. So we'll assume that or hypothesize that like that particular climatic event had an influence on the divergence or on the speciation events of the lines. And then objective number three is whereby like we'll be trying to understand the genetic structure and assess the genetic diversity because we've got like these disjunct populations. So we want to see if like those populations are isolated or they still gene flow like between the different populations. And then the last objective is whereby like we'll be trying to develop niche models to actually like try and predict how future climatic changes are actually going to affect the current distribution or the current species ranges that we have. So those are the four objectives, and then like I will talk about objective number one. So basically, I will try to combine objective number one and objective number two because basically, like the phylogeny that we're going to construct in objective number one is going to be the baseline for actually like dating and understanding the biogeographic patterns of Monopsis. And then like for objective number one, we've got like specific aims whereby we're just trying to understand the origin so that we can be able to actually like test hypothesis one. And then also Monopsis is a weird group because it hasn't been studied much. So we'll be trying to understand the relationship between like species. Because according to Peter, who's like a curator at Paris Herbarium, one of our collaborators, like some of the species, they look like hybrids, but no research has been done to actually like confirm that. So we hope that like doing like a phylogeny and the systematic like analysis will actually help us to understand if like we do have hybrids or we not. And then like after that we're going to actually like estimate the divergence time, as I've said, to actually like correlate like geological events and speciation events. And then after that will also be like hypothesis number one, where we're going to understand the migration pattern so that we can see if like Monopsis actually originated in Cape, like in the tropical forest and then migrated downwards towards Cape. So like those are the aims of like objective number one, specific aims of objective number one and number two. But we're not the first one to actually like use Monopsis. Previously, our postdoc in our lab, Crow, like actually like included Monopsis in his phylogeny. And then they found out that like Monopsis diverged about like 20 to 30 million years ago. And then like that actually like raises our confidence when it comes to our hypothesis number one, because previously they said like when you're looking at the Cape to Caroline hypothesis, they assumed that like actual radiation happened like in the Cape region or in the Mediterranean region. But the Mediterranean region is pretty new because it's about like late Miocene. So when we are looking at this phylogen, you can see that like the divergence of Monopsis happened like about 20 to 30 million years ago, which is somehow older than like a Mediterranean region in the Cape. So basically like, and then the tropical forest is pretty old. So we'll assume that actually like Monopsis originated in the tropical forest. But we are going to test that and actually like understand if like our hypothesis, if we can fail to reject our hypothesis. That's it. And then like for the methodology, I'm not going to talk much about it because it's going to confuse a lot of people because I'll just like go through it because yeah. So for sampling, I think like sampling for everyone, like whether I think like ecology or whatever kind of research, I think that's the most important part of like research, because with like adequate samples, we are able to develop like robust results. But if your samples was biased, if your sampling was somehow not good, chances are everything is not going to be like good, especially when it comes to results. So we'll try to use, or we're currently using like herbarium specimens, because logistically it's almost impossible to go to every like African country and collect samples. We can try our best, or we're currently trying our best to go to those countries that we can. If it is difficult for an African, like, so it's kind of hard. But like we're using herbarium specimens, and luckily enough, we've got like the Missouri Botanical Garden, which has like a lot of specimens, and then we also have a collaborator in Paris, so who's also sending some samples. I'm also from Africa, so I have access to like Sanbi, which is in Pretoria. So like they've been able to get like some of the specimens. I think right now we've got like 450 or 60 specimens from these two herbaria, and then we're still like waiting for samples from Paris. And then I'm also like doing my fieldwork. I just recently came back from my fieldwork in Swaziland and in South Africa, where I'm still like collecting samples so that we can have like adequate samples, and then we can be able to have like a robust like phylogen so that our results can make sense. And then for sequencing, I was just like we just because I don't have to go deeper into this, but like we'll be using nuclear markers through like target sequencing, and then we already like have process that we developed for Campanulaceae, this family, and then we're also going to like use process of AGS1353. And then the phylogen, like luckily in the Florida Natural Museum, we've got like Ortiz, like one of the lab technicians. It developed like a captures pipeline, which we're actually going to use to actually like do the phylogenetic analysis, and then like the trees will be generated using IQ3 and Astra3. And then for dating and ancestral reconstruction, we'll be using TPL, because we cannot use Bayesian methods because our data set is going to be a bit bigger. And then we're going to use like BioGeoBS to actually like reconstruct the ancestral areas. And then, but if you have questions about the methodology, you can ask me later. I can explain it to you like in details. And then so far, like as I've said, I've been able to go to like Pretoria to sample tissue and then like a botanical garden. But someone can ask that like herbarium specimens are pretty old. Some of them are 50, 60 like years old. How are you able to even get DNA? So it depends. Some specimens are good, some specimens are terrible. So like when you're going to the herbarium, basically you try to choose like specimens that are pretty good, whereby you can be able to get like some DNA. And then like we have optimized like a DNA extraction protocol, which actually helps us to get like DNA from all specimens. I think all the specimens that I worked with was like 50 years old. So, but we're still able to get like good DNA which can be used for analysis. So the herbarium are very important for like actually like storing the materials, because we use them as gene banks. So, and then like we're able to extract like DNA, and then we got quality DNA, and then like right now we've sent it for sequencing, and then we're waiting for the results, because sequencing also takes time, can take 3 months, 4 months, depending on the sequencing company. So far we've done that, and then we're still waiting for our reads. And then once we get our reads, we're gonna run do like the downstream analysis to actually like reconstruct the phylogen and run and do all like the other molecular analysis. And then for chapter 3, this is the most interesting chapter. This is the chapter that I like the most. So basically, like we'll be understanding like the gene flow between like the different like populations. I think like when you remember like the slide that I showed, but like you see that like there are different distinct populations of Monopsis. So we're trying to understand if there is gene flow between like these different populations, so that we can be able to identify like potential like gene flow barriers and then gene flow like corridors. And then we're going to identify species that are at risk of extinction, because if we've got like an isolated population and then there is no gene, there's genetic diversity or there's no gene flow in that population, chances are if like there's a random change in climate, like in climate events, chances are there might be like a population bottleneck and then like we might lose that population. So like this one is very important to actually like identify like those evolutionary units that are important, or those evolutionary units that actually need like immediate intervention so that we can be able to save them. And then the methods, the first part is the most interesting one, the one of collecting samples, because that's when I get to actually go to the field and then like do field work, collect samples, and it's fun to go to the field, but it's very fun. It was my first time going to the field, but like I really enjoyed it. And then like so I'll be collecting the samples that like are rare, like especially like in Southern Africa, because that's where I feel safe. In other countries, if the opportunity arises, I might go, but like right now I'm focusing on Southern Africa. And then like we're using structure analysis. I don't want to go deeper into that, but like basically like once you use structure analysis, it actually tells us the genetic like structure of like the different population, and that in such a way that we can be able to see if like there is gene flow between like isolated population or there's no gene flow. And then like the main focus like for this particular chapter is 3 species. So there's Monopsis zaheri, which you can see that like from that like a map, it appears in Eastern Africa, in Western Africa, which is like Angola, and then in East Africa, which is Tanzania, and also it occurs like in Southern Africa, if I'm not mistaken. And then there's also Monopsis delaroiti, which is occurring like in Western Africa and then in East Africa. But it also is another subspecies which is occurring in Southern Africa. And then I've got Monopsis simplex, which is kind of weird because it only occurs in West like Southern Africa and also like in Eastern Africa. So it will be interesting to see if like these two disjunct populations are still the same species, or if there is still like gene flow between these two like disjunct populations. So that's why we'll be trying to understand if there is gene flow like between like the disjunct populations, or there's no gene flow, so that we can be able to identify or actually like realize that okay, this is a particular isolated population, which means like there is no gene flow like between the different populations. So that's the main aim of like this chapter. And then progress. I went to the field for my first time going to the field, and then like it was tough at first, and then like I got used to it. So I was able to identify like some of the species and then like collect some of them, like which is like this Deciphering, Malvasia, and Stellarity. And then like what is tricky about these plants is that like they're small, as you guys can see. And then like in Africa we've got like grasslands, like the savanna grasslands. And the grasses they grow taller than the plant. So if the plant is more like this, so chances are sometimes you have to like, try to crawl, actually like look at the plant. But the tricky part about it is that the flowering time, we do not have like a specific flowering time because like sometimes you can go there and like when they are not flowering, it's kind of very hard to actually like find them and catch them. So that was a tricky part. Since I'm not a taxonomist by training, at first I thought that like maybe I'm the problem, I cannot actually like see the species. But then I went and brought like an expert and then we started with him. That's when I said like, okay, I'm not the problem, it's fine. So like the process in the field, I'm just gonna explain like the process like that we do in the field. So step number 1 from that picture on the left, you can see like a young handsome man walking under the. So basically like you walk. First we use like iNaturalist. I think everyone is familiar with iNaturalist to try and locate like the localities, and then you go to that particular locality and particular area and try to walk around. Basically, that's the only thing that you can do and to search for the plant. And then like once you identify the plant, that is like picture number 2, and then like you actually confirm if it is the plant because like some plants are almost similar, and then like after that you collect the plant and then like we use silica gel to actually like make sure that we dry it without degrading the DNA, because it is pointless to go and put the plant in the DNA like degrades, which means we just like wasting resources. And then after that, this is like one of like my samples from Africa, which I think was collected on 15th January. So like you can see that it is still green in color. So like you still can dry it, it actually helps to maintain the green color, and that way we preserve the DNA. So that's what we do. Oh, and there's like some picture that I took from the fields. Like this little mountain, it's in soil, and it's a beautiful. It's called as Bebero mountain. And also, like when you're doing research in Africa, we don't need money for lunch. Like when you go in the field, like there are a lot of fruits, so you just like be like the Asians, like get us and collectors. You collect fruits, you eat. So that's the most fun part about like doing research. So you guys encourage you to go to Africa to do research there. You won't need money for lunch. You just go and get it. Then like objective number 4, the last objective for them. So we're doing like nature modeling, which is not easy. I think like those guys who are familiar with nature models, it's not easy because like of the absence and present here, which complicates everything. But like we'll try our best actually like to generate species distribution models on selected species that we think that they might be at risk of extinction. And then like we're going to predict those species, and then like after that we're going to be able to infer conservation strategies. And then like we'll be using like data from like GPIF and iNaturalist, and also the data that I'm going to Stephen and down there. And then like the other variables like climatic variables will be downloaded from the major databases like Harmonized Diversity and all the other like online databases for climatic like variables. And then we'll use Maxent to actually like develop the models, but we might change it depending on our data set. And then we are still trying to collect like more geographical data because like we just want to make sure that we've got like ample data so that we can be able to develop robust like models. It's tricky, like developing each model is tricky, like the abstract data. So when we're going to the field, basically, like, that's what we do because like we decided to change the sampling strategy instead of just like recording like presence or the data that we found. If we go to a particular page and do not record a species, we just like verify that like we don't have like the occurrence, or we don't have the species at this particular time. So we're trying to change like the sampling strategy so that we can be able to have like present data in like a confirmed absent data set. And then we are currently about to run bioinformatic analysis when our rates can come back, and then develop the niche models. We are currently like on the verge of developing them, but we're not going to develop like the full data, like the final models. We're just going to trial them out using the available occurrence data. And then like once I go to the field and collect more data and to try and improve the sampling, that's when we're gonna try and create final niche modelings. And then like we're going to also run the population genetic analysis. And once I have adequate results, I'm going to come back probably next year around this time. So you guys expect me again to come back, and then like I'll be able to give more results about my research. So basically like that's what like I am doing, and then I hope that I will be able to actually like achieve that through your help. And then because I know that like we've got like a lot of experts. Some of you guys are already like familiar with African ecosystems, so I think like your assistance, like maybe questions that I overlooked, can actually like help me to actually like try and understand like more things that they can do to actually like answer the questions that I have. Yeah. I have to acknowledge some people, but like, yeah. So I've got two types of acknowledgement: people that I work with, and then like people who are funding me. I've got like less people funding me. I don't know why, but yeah, the Florida Museum has been like funding me since I came in. So because I went to Missouri, they were the one funding me. Then I went to South Africa, so they were the one funding me. And then like Sandberg, like it's my home because that's where I get most of the specimens. And then Missouri Botanical Gardens, same case, Paris and University of Swatin. And then like I've got that big team there, consists of my lab mates and my mom, who is the smallest, I guess, in that family. Yeah, that's my advisor. The smallest one is my advisor. She's my mom, basically. Like, yeah, she has been supportive so much. And then, like, lastly but not least, I want to thank the one and only, this handsome young boy. Like, yeah, he's like spearheading like the research. And then I forgot to include, yeah. And then I would like to then like to see the actually like giving me this opportunity to actually present. It's my first time presenting in a seminar. That means a lot to me. And thank you so much.
Great. We have time for questions, so we are up